Proteins. They make everything in our body from our muscles, skin, hair, and yes, even our fingernails. Let's find out how they're made. Hey, this lesson is on protein synthesis, or the transcription and translation of DNA, RNA, and forming proteins. So, the genetic code is determined by the arrangement of our nitrogenous bases within our DNA. And genetic code is actually going to code for the production of a specific protein. So, we have our DNA. We're going to transcribe it into RNA. And if we think about what a transcribing is, you're just rewriting something. So, we're going to rewrite our DNA into the form of RNA. Then from RNA, we're going to translate that into a protein. And again, when we think about what translation is, that's taking something and turning it into a different language. So we're going to take it from the nucleotide language of RNA to the amino acid language of a protein. So proteins are composed of 20 different amino acids that are strung together in endless varieties of combinations. They make up our cell membranes, organelles, muscle fibers, hair color, enzymes, antibodies, hormones, bones. All of that is made up of proteins. And it takes 3 different nucleotides, which is known as a codon, to code for 1 amino acids. And humans can make 12 of the 20 amino acids, but we must consume the other 8 amino acids because they're known as essential amino acids. We must get them through our diet. So a simple protein has roughly 8 amino acids, and we can have complex proteins up to 50,000 or so amino acids. And the sequencing of these amino acids is determined by the letters in our DNA. And even if we replace a single letter in our DNA, which replaces a single amino acid in our DNA, it can change the structure of that protein. But to protect against this, the genetic code has 3 important characteristics. One of them is the genetic code is redundant. So there's more than one codon that codes for the same amino acid. The genetic code is continuous. So it reads as a series of 3-letter codons without spaces, punctuation, or overlap. And the genetic code is nearly universal. So almost all organisms use the same genetic code, which is good for scientists when they're studying genetic engineering and biotechnology. So for the role of protein synthesis, DNA runs into a little bit of a roadblock because DNA is in the nucleus, but protein synthesis occurs in the ribosomes in the cytoplasm, and DNA isn't able to get out of the nucleus. So a carrier molecule, mRNA or messenger RNA, is responsible for reading the information from the DNA and carrying it to the ribosomal RNA in the cytoplasm to be translated into an amino acid. So mRNA is able to leave the nucleus and travel to the ribosomes in the cytoplasm. So RNA is different from DNA in the sense that it's single-stranded and is able to get out of the nucleus in the cell. It contains the sugar ribose instead of deoxyribose. It's located throughout the cell, where DNA is also found in the nucleus of the cell, and the mitochondria incidentally has its own DNA. It contains the base uracil instead of thymine, and there are 3 types: mRNA, rRNA, and tRNA. Messenger RNA, ribosomal RNA, and transfer RNA. And it's shorter. There's no introns. There's no part of RNA that doesn't code for proteins. So protein synthesis begins with the transcription of DNA into RNA. So we're forming RNA from DNA. And again, DNA is never able to leave the nucleus. It's protected. And in its double-stranded form, it stays where it is. So in transcription, the DNA molecule unzips, just like in DNA replication. However, RNA nucleotides are now added by RNA polymerases. So instead of using a DNA polymerase, we use an RNA polymerase because we're gluing on RNA nucleotides. And for each gene, only one strand of the DNA is transcribed, and this is called this sense strand. The other strand is called the antisense strand, and we're not going to code on that one. As the DNA double helix uncoils, nucleotides from the mRNA find their appropriate pair, their complementary pair, using the DNA as a template. And again, we're going to go from that 5' to 3' continuous direction. Uracil is going to bind to the exposed adenine bases, and cytosine bind to the exposed guanine bases, and vice versa. So the mRNA now will be joined with the RNA polymerases and fused into a long chain. Then in its single-stranded form, it's going to move away from the DNA, and the DNA will rejoin its hydrogen bases. The mRNA is going to leave the nucleus in search of ribosomes. So here again, we have that replication bubble, but now we're going to be using RNA polymerase to attach the RNA molecules onto our DNA in the complementary pairs. You'll see in that red strand that we have the nucleotide uracil instead of thymine binding onto adenine. So here again, we can see our complementary strands where the adenine binds to uracil, the guanine to cytosine, and any thymine in the DNA will then bind onto an adenine RNA molecule. So the mRNA reads the code from the dRNA, and the RNA is going to then code for different amino acids. Some codes in the mRNA are not for an actual amino acid protein, but are terminators and initiators. Terminators end protein synthesis, and that's known as a stop codon. So when our RNA code comes across a stop codon in the ribosome, no more amino acids are joined, and that protein is now complete. Initiators turn on protein synthesis, also known as a promoter site, and this starts RNA transcription. So if there was any DNA that was transcribed into RNA before the start codon, no amino acids are going to be attached until the start codon appears in the mRNA sequence. So here we have the methionine amino acid, and it would be AUG is going to be our start codon, and this is the only one. But we can see up in that first row that there are three different stop codons, UAA and UAG. So we read the first base on the left column, the second base around the top, and the third base down the side. And when you're just taking a quick glance at this chart, which is in your data booklet, you're able to easily see that there are lots of different mRNA codons that code for the different amino acids. Start is isolated and it's its own one, but everything else has at least two different codons that code for the same amino acid. So when we look at this example of a DNA sequence going through transcription and translation, we can see here in black what the original DNA sequence was and how it has thymine instead of uracil. So writing our complementary DNA sequence as we would during DNA replication, we would complementary pair the thymine and adenine and the cytosine and the guanine. Now, writing an mRNA sequence, we're going to refer this one back to the original DNA sequence. So where we have adenine, we're going to add a uracil. Where we have guanine, we're going to add a cytosine. And we can see that they're separated in little groups of 3 to represent our mRNA codons. So if we have 21 different nucleotides, we're going to have 7 amino acids. Now, looking at our amino acid sequence, what we're going to do is we're going to take our mRNA codon, and let's look at the first one, UUA, and go back to this mRNA codon chart, and we're going to go look at the U in the top column, the U in the first row, and the A third one down. So we have leucine. So then we would write the abbreviation for it there, and we would continue to go on through our amino acid sequence for the other 6 nucleotides. So arginine, serine, proline, serine, valine, and phenylalanine. So translation is that stage where we take the mRNA and turn it into amino acids. And this involves translating codons found on the mRNA into a chain of amino acids to form the protein. And another type of mRNA, tRNA, is an important part of this process. So tRNA is made up of a single strand of RNA that's folded into a cloverleaf shape. So one lobe contains the anticodon, so 3 nucleotides that are complementary to the mRNA codon, and at the opposite end is a binding site for an amino acid that corresponds to that codon. Ribosomal RNA is a linear strand of RNA that remains associated with the ribosome. So this one doesn't travel through the nucleus or the cytoplasm to get any free amino acids. So the exposed bases on the tRNA are known as the anticodon. These are going to bind temporarily onto the mRNA and hold the different amino acids in place before they can be bound together. So glutamate, which is carried by a tRNA molecule that carries either a CUU or CUC anticodon, which are opposite to the mRNA ones, which would be GAA or GAG. Valine is carried by a tRNA molecule that has the CAA anticodon, meaning the mRNA would be GUU. Here we have an image of how the tRNA temporarily binds to the mRNA. So it's that cloverleaf pattern. We can see where those nucleotides are together and complementary at the bottom. We can see that cloverleaf shape of the tRNA. And then you can see at the top that we have the amino acid. Now, again, these anticodons are going to be important because then as the next amino acid is carried up by a tRNA molecule, those amino acids will be held next to each other so that they can be bound together in a longer amino acid chain. So for our steps of translation, translation is activated when the mRNA molecule binds to an active ribosome complex in such a way that the two codons are exposed. And this happens in the ribosomes in the cytoplasm. The first tRNA molecule carrying the amino acid methionine or START base pairs with the START codon of the mRNA, which would be AUG on your mRNA codon chart. Another tRNA molecule arrives at the codon next to the first tRNA, and the first tRNA passes its amino acid onto the second tRNA, and those amino acids are now bonded together in a 2 amino acid chain. So enzymes are going to catalyze the formation of a peptide bond between the amino acids, holding them together. As the ribosome moves along the mRNA strand, one codon at a time, tRNA molecules will detach from the mRNA, and another tRNA molecule will come and attach, carrying its own amino acid, and then they'll be bound together. This process is going to continue until we come across the presence of a stop codon, UAA, UGA, or UAG on the mRNA. And the sequence of amino acids was originally described, carried by the mRNA from the DNA. So all of these proteins that are being coded for came originally from the DNA nucleotides. Okay, so here we have our tRNA, which is yellow, carrying the start codon in the top scene. Then in the next one, another tRNA molecule binded to the mRNA, and then a peptide bond is formed between the 2 amino acids. As that third tRNA came in, a peptide bond formed between amino acid 2 and 3, and that first tRNA, now that it's empty, is able to go and collect another nucleotide in the cytoplasm as the fourth tRNA comes. And this process continues until a stop codon ends the sequence. So lastly, we're going to look at this quick example. So we're going to write the tRNA and amino acid sequence for the following DNA. So our DNA we have here in black. And to go from the DNA to the tRNA, in between we have to put the mRNA. So in red, I have the mRNA of AUG, which is our start codon. And then our matching tRNA in blue would be UAC. DNA CAC in black, mRNA GUG in red, and then tRNA CAC in blue. Now you might actually start to notice a pattern here that the tRNA and the DNA have roughly the same nucleotide pattern. The only difference is the DNA has a thymine nucleotide, where the tRNA has a uracil nucleotide. Other than that, if we look, for example, in the second DNA column with CAC, the tRNA is exactly the same because we have that complementary strand of mRNA in the middle. So you can either go through both of the steps to go from DNA to mRNA to tRNA, or you could replace the thymine with uracil in the DNA, and that will get you your tRNA molecules. I hope you enjoyed today's lesson on transcription and translation. Catch you next time. Bye.